first session. So before we get started, we will have Dr. Mark Lipsich, the director of the Center for Communicable Disease Dynamics, to say a few words. Welcome, Mark.
Very few. Welcome to what I hope is the most chaotic with monotonically decreasing pattern of our seminar series. And a big thank you to Shweting, Kea, Joshi, and Nicole Kogan for organizing a really diverse and interestingly mixed set of speakers for this semester. I hope you'll all come back and bring your friends and even your unknown people you see around to the future seminars. And with that, let Shweting introduce our speaker.
Thank you, Mark. For a few logistics is that we will take questions after the talk. And for online folks, please send your question to Bradford Taylor. We're going to collect your questions at the end. So now I'm very excited and honored to welcome Dr. Gerry Tonkin-Hill as our speaker today, who is at the University of Oslo. Dr. Tonkin-Hill's research at the University of Oslo focuses on microbial genomics and the development of computational and statistical tools to track the evolution and transmission of pathogens. In particular, Gerry is interested in how pathogens evolve within the host and the role of horizontal chain transfer in the evolution of bacteria. He has developed several popular tools to infer pathogen population structure, transmission networks, and bacterial genomes. Gerry completed his PhD at the University of Cambridge and the Wellcome Sanger Institute, where he remains as a visiting research position. He is constantly working with the researchers in Australia and Cambodia on the impact of antimicrobial treatment on bacteria within host genetic diversity. And his talk titled today is Within Host Evolution and its Implications for Pathogen Genomic Surveillance. Well, thank you so much for coming, Gerry. Please take away.
Yeah, thanks very much for having me. It's really nice to be here, and thanks for introducing me, Shweting. Yeah, you've done a great job of covering what I was going to do. So my first slide was going to describe what I do, but that's kind of already being covered. But essentially, I'm really interested in looking at microbial population structure, and as part of that, looking at horizontal gene transfer between different bacteria. And what I'm going to talk to you today about is also looking at within host diversity and how we can use that to do genomic surveillance. So hopefully by the end of the talk, you'll have, I'll have convinced you at least a little bit, but by looking at within host diversity, we can do a better job of using genomics in surveillance of pathogens. So just first, what do I mean by within host diversity? Well, there's a couple of different main types we can talk about. One is looking at multiple colonization. So this is when you have two very distinct lineages. You could be colonized with them at separate time points, or it could be, say, a large bottleneck and you get two, but they're very different members of, in this case, I'm talking about the same species. The other kind of within host diversity we can discuss is de novo diversity. So this is where you get a single lineage or strain, and then you develop mutations within that carriage event, within that infection. And so throughout this talk, it's going to be kind of split into four main sections. The first one's going to be like a quick summary on why genomic surveillance can be useful. And I'll just introduce the data sets that I'll use as examples throughout the talk. I'll then finish the talk going back to genomic surveillance, but go on much more in-depth look at how we can use within host diversity and transmission. And I'll introduce our new tool called Track'em. And then as in between those two things, I'll discuss how by using genomic surveillance within host diversity in genomic surveillance, we can also look at things like the impact of antimicrobial treatment on diversity within the host and how different lineages compete within the host. So what do I mean by within host diversity? In general, I'm talking about when you look at genomic diversity using deep sequencing, so deep whole genome sequencing. But rather than metagenomics, where you sequence everything that you see in a sample, most of what I study focuses on things that have been enriched. So either by culture with selective media, you can enrich for certain bacteria, or you can do amplicon-based sequencing, as we often do for viruses such as SARS-CoV-2. So the two data sets that I'll discuss today, the first one is bacteria, Streptococcus pneumoniae, and this kind of was the focus of a lot of my PhD work. And then I'm sure we're all very familiar with the second one, SARS-CoV-2. And we had some early data during the pandemic to look at within host diversity of kind of the initial outbreaks of SARS-CoV-2. And so the bacterial data set I'm going to discuss is from the Maela pneumococcal deep sequencing study. And so this is based on some data that's actually quite old now. It was collected between 2007 and 2010 from a refugee camp called Maela on the border of Thailand and Myanmar. And this refugee camp is mainly made up of people, ethnic Karen people who've fled persecution in Myanmar and ended up in border camps along the Thai border. Initially, this study involved the recruitment of just under 1,000 pregnant women from a camp with an overall population of around 40,000. So it's a fairly large camp, but the size of the camp is still quite restricted. I think it's at its largest distance is about 4 kilometers in length. But crucially for this study, movement in and out of the camp is quite heavily restricted, and the administration of antimicrobials to people within the camp is very carefully monitored. So we have a pretty good idea of when people take antibiotics and exactly what they've taken. So just a little bit more detail about how the sampling was done. Once the women were recruited, when they gave birth, their children had nasopharyngeal swabs taken every month at their monthly checkup for the first 24 months of their lives. And a subset of the mothers also had monthly swabs taken. So this led to around 19,000 swabs being taken, of which around 11,000 were positive for strep pneumo. So there's very high prevalence of the bug within the camp. And 4,000 of these we sent for deep sequencing. But so just to go into what I mean by deep sequencing in this context, kind of the top bar here is what we would do in a normal genomics pipeline, where you would take a nasopharyngeal swab it out on a selective agar that selects for strep pneumo, and then you'd select a single colony from that and re-swab it out on another plate to enrich for that colony. So by doing this, you're kind of focusing on just a single representative genome. We skipped the second step, so we just took the first selective agar and sent the whole thing for sequencing. So by doing this, we're hoping to enrich for all the strep pneumo within the sample and not just a single gene. The COVID data set I'll be talking about was from quite early on in the pandemic, so from March and April in 2020. And these samples were the ones that were sent to Addenbrooke's Hospital in Cambridge, UK. But they were from people that were kind of from all over the southeast of England. And so overall, we had over 1,000 samples in this data set, and we wanted to look at the within host diversity of these samples. And so the way we went about this was the kind of pipeline that was being used at the Sanger at that time was Amplicon-based, but we wanted to make sure we were looking at real within host variants and not errors. And to kind of get at this, we ended up doing every sample in technical replicates. So we performed separate reverse transcription, PCR, library prep, and sequencing steps, and then only considered within host variants that were found in both of those technical replicates. So the first kind of major point I'm going to discuss is multiple carriage. So here we're talking about when you have two fairly distinct lineages of the same species. And we found that by doing this deep sequencing-based approach on the pneumococcal carriage, we could identify 23.6% more serotypes or lineages in this case, compared to latex sweeps, which is kind of the gold standard for looking at multiple carriage in strep pneumo. So this approach is much more sensitive, and kind of along with that, as well as just identifying the serotype, which is all that you can see when you do latex sweeps, we could also look for resistance elements. And so using this approach, we identified just around 14% more resistance elements than if we'd just taken a single representative genome from each of these samples. But probably most interesting was that when we looked at the frequency of the lineages within these samples, they weren't randomly distributed between the different lineages. And what we found was that lineages that are known to be invasive, including serotype 1 and serotype 8 genomes, were often found at much lower frequencies within the host. So you were much more likely to miss these more invasive lineages if you were just taking a single representative genome in this kind of surveillance technique. So we also looked at multiple colonization in SARS-CoV-2. So this was early on in the pandemic, so prevalence within the country was quite low. And obviously, the overall prevalence of the bug has a big implication on whether or not you're going to see multiple carriage. But despite this, we still found evidence of multiple carriage in around 3% of our samples. And to just give you an idea of what this looks like in our data, essentially on the x-axis here, you've got the position in the SARS-CoV-2 genome, and then you've got the frequency of variants on the y-axis. And you can see that there's one kind of band at about 10%, which is the kind of minority infection. And then you've got the majority infection, which is up on the blue band at the top. And we looked at whether these variants matched with known lineages that were circulating in the UK at the time, and they matched pretty well. And so it's a bit hard to see here, but there's a couple of grey variants, and they were the only ones that didn't match with known variants. So the two bands matched very well with what we thought was circulating in the population. I should say that most of these samples were taken from when people were in hospital. So it's possible that some of this co-infection was occurring after they were kind of already grouped together. The second thing we can look at, so other than just looking at multiple carriage, by doing within host diversity, we can also look at how things evolve within the host. And so one of the major things we might be interested in is selection. But more importantly, what we were interested in is whether there were different selective forces acting at different time scales. So within the host versus over longer time scales between people. And so to do this, we looked at DNDS, which I'm sure a lot of people have already seen, but just for those, just as a reminder, I guess, it's just the ratio of synonymous or silent mutations that probably don't have a phenotypic effect to non-synonymous mutations. And these can include nonsense mutations, which essentially knock out the gene probably, and missense mutations, which might alter the function of the gene. And so we're kind of looking at whether the ratio is below 1, which would indicate purifying selection. And what we found was that in SARS-CoV-2, there was strong purifying selection within the host, which is what you might expect, but that it was less strong than what we observed between hosts. So there was less selection over this very short time scale than we observed over longer time scales between people. But when we looked at these variants at what frequency they were occurring within the host, very few of them were getting it to a higher frequency. So once you looked at which ones were actually getting to higher frequencies, it looked a lot more like what you see between hosts. So these variants probably don't last long and get knocked down quite quickly. We could also look at this in strep pneumo, and interestingly, we found very, very similar DNDS ratios for pneumo, which is, I guess, consistent with them being located in similar places within the human respiratory tract. Because we had more samples, we could also look at whether this DNDS at the gene level, so whether there was different selective forces acting on different genes. And what we found, interestingly, was that the strongest purifying selection was acting on genes involved in the pneumococcal stress response, which it's maybe not straightforwardly obvious, but if you can look at genes that are under strong purifying selection and identify those that are, identify targets for those genes, so targeted treatments for those genes, then it might be harder for the bug to evade those treatments if they are under strong purifying selection. So this is the genes we identified in strep pneumo. I don't have a great target for any of these genes, but this is why this kind of analysis might be interesting. In addition to selection just occurring in people in the absence of treatment, we can also look at what happens when you give them antimicrobials and how this affects selection within the host. And to do this, this is again looking at the pneumococcal example. We looked at children who'd been treated with antibiotics, and we had samples before and after they were given the antibiotics. And we also had a control group where there was a similar time between the samples that had been taken, but the child had received no antibiotics. And we were looking at whether there was a difference in the prevalence of resistance elements or other elements in children after having received antibiotics. And what we found at the lineage level was that children who'd been treated with antibiotics were much more likely to have a dominant lineage that was the multi-drug resistant lineage, GPSC1, or Global Pneumococcal Sequencing Cluster 1. And a lot of this was driven by the fact that the children were colonized initially with multiple lineages and one was susceptible, and then the antibiotics killed off that one. But even once you controlled for this, children were more likely to be colonized after having been given antibiotics. So receiving antibiotics in this camp meant that you were more likely to then be colonized with GPSC1 subsequently. Aside from looking just at the lineage level, we could also look at the level of individual mutations within genes and look at whether there were certain mutations, whether certain genes were enriched for mutations after being given antibiotics. And when we did this, the genes that popped up were the well-known penicillin binding proteins. They're very well-known drug resistance mechanisms in strep pneumo, as well as the ABC transporter protein, which has also been previously associated with resistance in pneumo. But what was kind of really interesting about this analysis is the vast majority of resistance phenotyping genotype association studies in pneumo are done based on laboratory techniques. So by doing this kind of surveillance, you can actually look at resistance occurring within the host without having to then subsequently do the laboratory-based analysis. And although we didn't identify any novel resistance mechanisms, if there was some kind of mechanism that was hard to reproduce in the lab, you might observe it by doing this kind of surveillance. So this is a slide aside, but we could also look at whether certain resistance lineages outcompeted susceptible lineages, or whether it was the other way around, so susceptible lineages outcompeting resistant lineages. And this is quite important for a lot of models of the dynamics of resistance in Streptococcus pneumoniae populations. And what we found was that resistant lineages tended to be found by themselves within a child much more often than we would expect, given their overall prevalence within the population. So they were more likely to be found in isolation, which suggests they're probably outcompeted by susceptible lineages within the host. So that was kind of the general genomic surveillance and how it can improve our ability to detect lineages and how it can also give us some other interesting information. And now I'll go a lot more into how we can use data on within host genetic diversity to look at transmission. So what are the benefits? Why would we want to do this? The first one's kind of obvious, and we've already covered it a little bit. But if you have colonized with multiple lineages, if you only take a representative genome, so in this example, if we only took the purple lineage in the right-hand person and the green lineage in the left-hand person, we might miss the transmission of the orange lineage. The second, probably more interesting benefit of using within host diversity is it can help us to get an idea of the direction of transmission. So if here we have a phylogenetic tree, and then in this example, we've got a single genome sampled from 3 people, there's actually 5 possible transmission scenarios that could all explain that single phylogeny. So in the second row here, we have the color of the phylogeny is the host in which the bug was in at that point in time. So in the first tree, we have that it started in person A, and then went into person B, and then transferred in person C. But the same phylogeny could also be explained if it started in person C and independently transmitted to A and B separately, and so on, and B and A, and so on. But if we start getting more genomes per person, we can start to disentangle some of these things. So if we observe that all the genetic diversity of person C was nested within person B, which was nested within person's A genomes, then we might be pretty sure that person A infected person B, who then went on to infect person C. Another thing we can look at if we have information on within host diversity is the transmission bottleneck. I won't go into this a lot in this talk, but essentially by looking at within host diversity, you can get an idea of whether transmission involves very few variants being moved across, or whether it's a very large bottleneck and you have most of the diversity from one person going on to the next. But that sounds great, but there's some significant challenges to doing any of this. So the main one that I'm going to keep going on about is that many sources of within host minority variants have nothing to do with transmission. And so just counting the number of shared within host variants is a very, you can run into a lot of problems if you're using that to infer how likely 2 people are to have transmitted to each other. So again, with the multiple carriage, but this time coming from a slightly different angle, you can have a scenario where 2 people share a minority lineage, but that lineage can have transmitted from 2 independent transmission events. So they can have a shared minority lineage that has nothing to do with transmission directly between them. Homoplasies is another big one. So here is if you have sites that are more easily mutated or hypermutating sites, then you can have the case where you get a lot of mutations recurrently occurring and very independently in multiple people. And then if you were to count those and look at them between people, you might incorrectly infer that they were the result of transmission. And so to just give you an idea of what this looks like in SARS-CoV-2, here we have the 1,181 genomes from before, and the tree on the left is the consensus phylogeny, so what we would normally do when building a phylogeny of SARS-CoV-2 genomes. And then on the right here, we have the number of shared within host minority variants between each of these samples. And so you can see that even up to 10 within host variants are shared over very large distances of the phylogeny, which probably involve many, many, many transmission events, and so are highly unlikely to have anything to do with transmission. And so this is probably the result of mainly recurrent mutations occurring independently, and also possibly some co-infections. With bacteria, as always, things often get a little bit more complicated. And so if you have different copies of genes in your genomes, you can also incorrectly infer within host variants. So in this example, you have a reference genome and it's got a single copy of a gene, but the sample you're looking at, the bacteria happens to have multiple copies of this gene. And when you align all your reads to the reference genome, you'll incorrectly infer that there's within host variation occurring at that gene. And this, a similar problem would also happen if you had a contaminating species that happened to share homology with the thing you were looking at. And to show you what this looks like, here's one of our pneumo examples. And so here we've just got to think of this as the reference genome. And here on the left-hand side, we have the frequency of within host variants along this reference genome. And you can see that there's huge clusters of within host variants, which essentially correspond to either gene duplications, or in this case, a lot of the time they were the result of phages sharing homology with the pneumococcal genome. And finally, a slightly more boring one, but we also have variable sequencing coverage. So obviously, if you only look, if you have very low sequencing coverage, you can't really look at within host variants, so you can get biases due to variable sequencing coverage along the genome. So what can we do about all of this? One way would be to come up with some filtering strategies. So with SARS-CoV-2, it's very well studied now, and so there's some pretty decent, I guess, filtered sets, so you can filter out variants where you're fairly sure things can go wrong. So Nicola de Mauer did this work fairly early on to identify a list of sites within the genome that could be potentially problematic. If we're looking at gene duplications and things like that, we can also filter out these areas that have elevated numbers of variants, so well above what we would expect given a normal evolutionary rate, and filter out based on that. So in pneumo, we did this and it worked fairly well, where you just have a fairly simple scan statistic looking for elevated numbers of mutations. And if you call an area as having an elevated number, you just filter those variants out. But this doesn't really solve all your problems. It doesn't help if you're looking at metagenomics, and in a similar way, it doesn't really help if you have multiple infections. And obviously, all of these things are very sensitive to how you're doing the filtering. So you have to be very careful about how you're choosing your filters. So how do we deal with this? Well, if we were just looking at single genomes, there's some complicated and quite informative methods out there for looking at transmission using single genomes. But then if we're having trouble with a lot of this kind of stuff, there's often SNP distance, which is a very simple but very robust method, and we often fall back to it when things are getting a bit difficult. But when we're looking at within host diversity, things aren't as simple. So we have some very good methods, but they're quite hard to run and require a lot of expertise and experience to run well. And there's not really a nice, easy method that could be used by public health officials to quickly rule out transmission. And so this motivated us to come up with a new method, which we've called TRACM, which roughly stands for Transmission Clustering of Metagenomics or Multi-Strain Samples. And what it does is it automatically selects the reference genome you should align to. So it decides what reference genomes should be within your sample. And it uses an empirical Bayes method to account for variable sequence coverage. Variable sequencing coverage along the reference genome. And then we have a rapid SNP and transmission distance estimation step that takes those estimates and estimates the likelihood of transmission between two people. And so what do I mean by the SNP distance? So just to refresh everybody, this is the classic SNP distance. So if you have your two single genomes, you just count the number of different places at which they differ or sites at which they differ. And the normal way we would use this for transmission is if the number of SNPs fell below some threshold, we would say that transmission was plausible, but not necessarily direct. Or if it was above some threshold, we would say that it's highly unlikely that these two genomes were related directly by transmission. To extend this to mixed samples, we wanted a lower bound. So we wanted to still be able to exclude the possibility of transmission using our method. But this, again, doesn't say whether someone definitely did directly transmit to someone. It just excludes the possibility that two genomes were separated by transmission. And so the way we went about this is, rather than just looking at the majority variant, we only counted mismatches if there was no common allele at that site. So in the first example on the left, you have two matching red alleles in this example at the consensus level, and then a minority allele that's a purple allele. In the middle, you have a mismatch where there's a red and a purple at the consensus level and nothing else. But on the right, you have a red at the consensus level and a purple at the consensus level in the other sample, but there's a corresponding purple at a minority level in the first sample. And so that one we would count as a match because there was plausibly a match between those two samples. So in this example, again, we've got a SNP distance of 1, but it's a lower bound. And to deal with variable sequencing coverage, we'll come back to the COVID example from before. So here again, we have the one lineage at 10% and one at 90%. And so to give an example of how this works, if we had 30 times read coverage at a particular area of the genome, we'd expect about 3 of those reads to have come from the red lineage. And so if we only saw one allele, say an A, we'd be pretty sure that the A occurred in both the red lineage and the blue lineage. But if at another site we only had 5 times coverage, we would expect all of those reads to have come just probably from the blue lineage, and we would say we didn't actually know what was going on in the red lineage at that location. And so we would call that site as ambiguous. And so we ran this on the strep pneumo samples from the refugee camp in Maela, and we found that of the close transmission pairs it identified, around 80% involved transmission from one of the 2 people had multiple lineages, which suggests in a worst case scenario that you could miss up to 80% of transmission pairs. Obviously, it's unlikely that you would always have the worst case scenario. And so you would identify some of these as they would both be at the majority level in both alleles. But the plot on the right is a map of the refugee camp, and the blue lines indicate transmission links that you would probably have picked up if you'd done traditional single genome surveillance, whereas the red lines indicate additional links that we found using this within host approach. So as well as looking at transmission around the camp, we're also really interested in transmission between mothers and their infants. And to get an idea of direction, we just took a really simple approach in this case, and we just looked at who had become infected first. So if a mother was infected with a lineage first, and then we identified a likely transmission link to their child that occurred later on, we would say the mother went on to infect the child. Vice versa, if the child became infected and then the mother, we would say the child infected the mother. And if they both became infected at the same time point, we would say that it was an ambiguous direction we didn't know. And just to give you an idea of what this kind of data kind of looks like, here we've got the sampling time points, and the colors indicate the lineage, and the black lines indicate likely transmission pairs identified using our algorithm. And so what did this identify? Well, I should say, we were also interested in whether this changed between when the child was less than 1 year of age and older than 1 year of age, because we're interested in whether the dynamics changed as the child grew older. And what we did find was that as the child grew older, when they were very young, they were more likely to be infected by their mothers than once they were over 1 year of age. So this suggests that the transmission from mother to child is stronger when the child is very young, and perhaps there's much more close contact between the mother and the child, and that this then weakens as the child gets older. An important caveat of this analysis is that we weren't sequencing other children within the household, so it's plausible that the other children were infecting both the mother and the child, or the mother and then the mother was going on to infect the child. But the fact that those children were also present in the second year suggests that the dynamics at least are probably changing a bit between the mother and the child, between less than 1 year and older than 1 year. So thanks very much. That was a bit of a quick tour about the kind of stuff I use within host diversity for. So hopefully I've convinced you that by looking at deep sequencing and deep population sequencing, we can do an improved job of genomic surveillance, and that TRACM, which I'm hoping to release publicly soon, is a useful tool to do this. And as well as allowing us to do genomic surveillance, it can also let us look at things like resistance dynamics within the host and identify important things like virulence within the host. And finally, by looking at within host dynamics, we can also look at the competition between lineages and the impact of drug treatment as kind of side things apart from just doing the central genomic surveillance main aim, I guess. And thank you very much for having me.
Thank you so much for the terrific talk, Dr. Tonkin-Hill. We're going to take questions from now on, and we're going to take as many questions both on-site and online. So if you have questions, online guests, please send your question to Bradford Taylor, and we will try to ask you to unmute yourself or read out the question for you. We're going to start with our in-person audience. If you have any questions, please speak up.
Yep.
It was great.
Thank you.
A few questions about the SARS-CoV-2 infancy. So I imagine the attempts to look at within host diversity and link people depend on what your expected bottleneck size is for infection, and then also when in the course of infection you're sampling. Given both population size that you're really sampling and the potential for diversification over the course of infection. And I'm curious how you account for those in, well, first, what are do you have estimates for both these processes, so bottleneck size and diversification, and how sampling at different times may inform your within host diversity estimates, and with those, how you take them?
Yeah. No, that's a great question. So the answer in general is it's really noisy. But we looked at whether the number of variants changed over time. We only had like relatively few instances where we had multiple samples over like the course of an infection to look at whether the number of variants was changing. And it was very weak, but there was a signal that it was increasing throughout the course of an infection. Sorry, what was the second part of the question?
Bottleneck size.
Oh yeah, bottleneck size. So I think.
Can you repeat the question?
Oh yeah, sorry. Yeah, sure. So the question was about bottleneck size. So I think bottleneck size is really challenging to gather. A lot of the reason is because you have these recurrent mutations. So if you're using that to estimate bottleneck size, you can easily estimate an inflated bottleneck. The other thing I've kind of been worried about is that I think bottleneck depends a lot more on how the transmission happened, necessarily more than like exactly what's happening with the pathogen itself. So obviously if you're kissing someone or you just walk past them, you have very different numbers of variants. And I think that increases the stochasticity a huge amount. So it's very hard to kind of infer, I think, a reliable bottleneck. So I didn't really attempt that, to be honest. And just concentrated on things that we needed to be very careful of when we were trying to do those subsequent analyses.
Any other questions? Yes.
Yeah.
That was great, Gerry. Absolutely was. I will talk to you about other things later, but when it comes to pneumococcal, that stuff about the serotype 1s and 8s, that was really quite interesting because, as we know, they are considered very virulent, but that's partially because they're so rarely isolated carriage. To what extent could your observation explain some of that? In the sense that they are more frequently carried than we think, but they are not as likely to be detected because there is something else there which gets flagged up first.
Thanks. So yeah, Bill was asking about the pneumo example and how we found serotype 1 and 8, which are invasive lineages at lower frequencies, and whether that could explain the fact that they're rarely observed in carriage samples. I think it could explain a little bit, but I don't think it's anywhere near enough. Like the difference we saw was substantial, but not enough to explain the difference in carriage samples. So I don't think it's just that we're not observing them because they're always at low frequencies. I think it's probably something else.
So what tighter are they carried out when they're on their own? You get more of the effect on their own.
I didn't look at the overall abundance of them, like aside from the relative frequency within the host. So yeah, I'm not sure. Yep, let's do it.
I was curious about the antibiotic resistance rates. So you say that resistant lineages were more likely to be found in isolation, even in the absence of antibiotic treatment.
Yeah, so ignoring antibiotic treatment, I think we only looked at samples that were well outside of the impacts of antibiotics in that analysis. And we found, the resistant strains were more likely to be found by themselves than with other strains.
Yeah, what do you think is going
I think they're probably just slightly less fit, but I don't think it's super strong. So we still found plenty of instances where it was likely that there was a resistant and a susceptible strain within the same host. So that you haven't got like really, really strong selection going on where they just push each other out instantly. But there might be a small selective advantage. That means that you're more likely to see one by itself. Yeah.
Just a question on the computational methods used in the project. How do you distinguish between strains you're doing deep sequencing and higher? Like, do you have enough diversity between serotypes that you can the same a given context? If you're doing something like relates to one serotype versus the other, do you have any ambiguous ones? How do you resolve this?
Yeah, so the question was how we separate the mixed samples into their different lineages or serotypes and then analyze those. We were very lucky. So this study has a previous study done on the same data set where they did single genome sequencing, and there were around 3,000 single colony picks in that study. So we had a very good idea or a good reference database for what should be in this population. And so we used a method called MGEMS, which essentially aligns to a reference database and has a probabilistic allocation of reads to different lineages, which is very reference dependent. But because our reference database happened to also be from the same data set, we were had a big advantage in kind of separating the samples out.
Sorry, follow up on that question. What about for SARS-CoV-2?
So I probably heard that, but how about for SARS-CoV-2? I'm much less sure about how easy it would be to do with SARS-CoV-2. Probably, I think you could do other methods based on phylogenetics more easily than the MGEMS approach, because it's much more dependent on the reads having multiple variants within a single read. So I'm not sure how well it would extend to SARS-CoV-2. I think up the back.
It's just like building up on the comments that the amount of variation might change with the course of the infection. I think that the diversity that's captured by a single sample at a single time point and in a single site, even though these are the examples of the, I mean, I don't know, maybe creating the but it might not be very representative of the whole spectrum of the diversity. So how do you think that impacts the results and so the need to be economic analysis?
Yeah, so the question was how representative is a single sample of the within host diversity over like a whole infection, is that correct? Yeah. I think it probably depends a lot on the bug and how quickly it sweeps through. So how quickly you get like sweeps to fixation of different alleles. In pneumo, the variants were fairly persistent, and in the small numbers of SARS-CoV-2 samples, we still observed the same variant multiple times at different time points. And so I think they are informative, but to really get an idea of selection and other forces going on within the host, you have to look at many samples. So even just looking at the same person with higher density sampling would be useful, but to really get a better idea of what's actually driving things, you have to look at many samples at once, I think. Yeah.
Yeah, so I got a couple of questions from Zoom. Somebody asked the more direct one first. Matt J. Sangor asks, have you tried using this approach, using long-read data?
So I think that's really interesting and I really like to, but no, I haven't. I think that would be super useful though.
Yeah. That was a short question. The longer one is from Peter Hertz, who's asking about wastewater sampling. Peter mentions that wastewater sampling is more and more important to identify variants during outbreaks. There's speculation that SARS-CoV-2 sequences in particular are from replicating variants in the gut. If that's the case, are there potential issues regarding making inferences about transmission as gut adapted strains could be diverging from the transmissible respiratory strains? There are differences between the two areas.
Yeah, so I think, I guess it's hard to infer transmission from wastewater sampling as a starting point, but I think you probably would get slightly different variants. This is just me guessing, really. But I think that brings up another point, which I didn't really make properly in the talk, which is that in the early stages of an outbreak, you might not know which variants are hypermutating. So I had that list of SARS-CoV-2 mutations that are pretty well known now, tend to mutate more often. But very early on in an outbreak, you might not have that. And if you're looking at within host diversity, you might be able to account for that more by being less prone to a single mutation changing. Yeah.
Great. I think for the sake of time, we're gonna stop the discussion here. On behalf of the Center for Communicable Disease Dynamics and this Harvard Chan School of Public Health, let's thank our speaker today for the please, so today's session is the end, but please do get in touch with our speakers if you have any questions unanswered or further interest on your research questions. Please feel free to reach out. And we're gonna have more seminar series coming for this semester. So please check out our website to stay up to date on the upcoming seminars. And hope to see you next week.
Thank you.